Good afternoon, everybody. I'm just going to talk about one of my studies using little animal models for Parkinson's disease. Okay, what a worm can teach us about Parkinson's disease. My focus is on dietary plant lectins and the mystery of causation. I'd like to thank for the institution's support the study, ACT Center, and Penington Biomedical Research Center, and the foundation, and also specifically I wanted to thank for the Raleigh Family Foundation for their generous and continued support on my research. What does Parkinson's disease look like under the microscope? The nerves that contain dopamine are damaged. The damaged nerves have something inside them called Lewy bodies we've seen earlier in Dr. Stanner's presentation.
Can you hold this close?
And the Lewy bodies that contain clumped, aggregated alpha-synuclein. Dr. Heckel-Breck had described where the Lewy bodies are in humans. They're in the nerves in the stomach, in the intestine, and even in the nose first. But then they show up in the nerves leading to the brain, and the nerves in the brain damaged in Parkinson's disease. And he proposed something enters the nerves of the stomach, intestine, and nose, and what it is has been a mystery. And this mystery agent travels in the nerves to the brain. This picture shows the Lewy body here, and this cell loses the normal structure of the cell. And Dr. Breck had found those Lewy bodies in the vagus nerve, in the stomach and intestine, and in the nerve system that innervates to nose. And he proposed this damage is in the ascending manner, going upward to the brain, and when the disease progressed, and lastly going to the highest brain and caused Parkinson's disease. So he has proposed there is a mystery agent that traveled along the nerve system, caused Parkinson's disease. What is the mystery agent? Although controversial, it has been reported that vegetarians may have more Parkinson's disease than people who eat meat. Lectins are abundant in the vegetables and the seeds people eat, which could be one of the many possible causes for Parkinson's disease. What are plant lectins? Lectins are proteins connected to a sugar. They are able to enter the walls of the stomach, intestine, and nose. The sugar attaches to the cells that may swallow the lectins. This property of lectins is used to improve drug delivery in the intestines to treat diseases. Lectins can be transported up or down the nerve fibers and cross nerve connections. This property is used to trace nerve pathways. Lectins help to deliver genes without using attenuated viruses in gene therapy. What we know about bad lectins? People in Guam, research has shown that people in Guam who eat cycad seeds develop Parkinsonism, dementia, and ALS. Lectins are found in cycad plant. Other studies have shown horses that eat yellow-starred thistles that contain bad lectins develop a disease similar to human Parkinson's disease. Why bad lectins could cause Parkinson's disease? Those lectins can aggregate alpha-synuclein, and they may carry other bad agents to enter the brain and damage dopamine neurons to cause Parkinson's disease. This picture showed this is a picture showing aggregated alpha-synuclein. This is in vitro. And when fed rats with fluorescence-labeled lectins, showing in the brain sections, those lectins labeled brain cells. So that means the lectins traveled from the GI system to the brain. And then we switched to C. elegans model because it's more convenient. And this can be used to study human diseases. I'll just go through those pictures to show the life cycle of the C. elegans. This picture is showing C. elegans under microscope. And then they had adult animals had lay eggs and hatch in several hours. Then they going through several stages. Within 72 hours, they reached adulthood. And then they can live about 20 days. When the food is not available, they kind of get hibernated. And then they can come out if the food is available. Normal worms is normally called wild type N2. And adult C. elegans about 1 millimeter long and round shape. They have 20 days of lifespan. They're transparent, make the study easier. They're both male and female and self-fertilize. They have total 959 cells and 305 neurons, and total 8 dopaminergic neurons. All of the genes in the worm are known, and 65% of them is related to human diseases. And this animal model does not need any regulations. I'm going to show you some research data. Lectins are transported to dopamine neurons in C. elegans. This picture showed an entire C. elegans. They're engineered to express green fluorescence in the 8 dopaminergic neurons. There are 6. Where is it? There are 6 in the head and 2 in the tail. We feed them with lectins, which labeled with red fluorescence, and this picture showing 7 out of 8 neurons are co-localized with the green fluorescence and the red fluorescence. So the co-localization can be seen as orange. And then when feed bad lectins, those lectins induced alpha-synuclein aggregation after transported to dopamine neurons. And this is green showing dopamine neurons. The red one showing labeled lectins transported to neurons. When you overlap those two pictures together, you can see the co-localization of orange-colored neurons. And then I used to test alpha-synuclein, see if we can see any of them there. And then we can see we use the blue color to label alpha-synuclein. And then when the 3 colors overlapping together, the white color showing here in this picture a white alpha-synuclein aggregation. Our research goals are to screen a library of lectins, to identify bad lectins that damage dopamine neurons and impair the ability of the C. elegans to move, to create Parkinson's disease model in C. elegans, which can be used in research to understand the mechanisms of the disease, and to develop new methods to prevent and treat Parkinson's disease. Working in progress, we have a proof of concept with our preliminary data. And I just received 3 years of funding from the Board of Regents to study on this project. And we have other projects to develop novel treatment on Parkinson's disease in collaboration with biomedical and agricultural engineering at LSU Ag Center. At the end, I'd like to thank for all the advisors, co-workers, student workers, and institutions. Dr. Yuan Luo, she's the one inspired me to use C. elegans study. Dr. Chris Jason Danner at the University of Louisiana at Monroe, Dr. Andrew Davis at Virginia Commonwealth University, they provided hands-on help on C. elegans studies. Dr. Jeff Keller at Pennington and Dr. Roger Lin at Department of Biology Science at LSU, they're advisors and provided insightful help and guidance. And my technician and student workers, they're all from pre-medical and pre-dental program, and a student in Louisiana Biomedical Research Network, all participated into this study. And Mr. Wing Chen Fan from IT department at LSU provided data mining. And also, I'd like to thank you for neurobiology research program, cell biology and cell imaging facility, comparative biology facility at Pennington Biomedical Research Center. Thank you very much.